Hey, Josh, how are you?
Hey, James. I'm doing fine.
Hey, Drew.
Hey,
I hope Josh, maybe we should send a reminder email to people. I'm
Okay,
sure
I'll do it.
everybody is overjoyed with the idea of another class meeting. Since it's not an official class time, if we could keep it a short meeting, if people, depending on what people have to present. Drew, did you have any luck getting it uploaded to NanoHub?
I'm trying to check right now.
And what about Slider? Well, you already had some of those things implemented already. So.
Yeah, I haven't made a whole lot of changes, but I'm trying to get it working as an app.
What are your plans for the summer? There's TJ. Well, this is quite an interesting ratio of instructors to students. Nothing wrong with that.
I'm outnumbered.
Well, Feynman said the best way to learn something is to teach it.
It's a reasonable point of view. I mean, I thought it was clear last week, people said they wanted to meet now. So it was,
That's was my understanding as well, but
Josh, did you include, maybe, is there an issue with the link?
it's the same one I used before.
Right, well. Let me just send it again. Actually, I don't want to do it that way. Yeah. Hey, Jeff. Well, give it another minute or two and then. TJ, do you know if anybody specifically was coming?
Yeah, both Tyler and Callie, we've discussed being here. Oh, there's Callie.
Okay, well, I guess maybe we should get started. Collie, would you like to tell us where you are with things and how it's going?
Sure. Just give me 1 second just to get everything ready.
Oops.
I can't share my screen while someone else is sharing their screen.
No, that's fine. Okay, well, let's get started then. Welcome to the extra meeting of the class, Engineering 443. As usual, we're streaming this, so let me know if you want me to turn things off. And This was an opportunity to do follow-up presentations, updates on where people are. So thank you for showing up. Holly, I'll turn it over to you now.
Okay, so this is sort of going to be a bit of an expanded update, giving some of the biological information that I have presented previously, but haven't really been doing the past couple of weeks because I didn't want to be redundant. And then once this presentation is over, I'll then share what's going on in CompuCell. Now, going back to the biological background, as you know, this project is modeling the end of the life cycle of a soil-dwelling amoeba called Dictyostelium discoideum. That's its scientific name. And this amoeba, once at the beginning of its cycle, when they're all different individual amoebae, when they're under starvation conditions, they send out a signal of a molecule called cyclic AMP, which you can make from ATP. And this signal causes the different amoebae to aggregate together and form a motile slug. And it's called that because, well, it's not truly like a slug like an animal. It's this aggregation of amoebae that can move together, hence it's a motile slug. So this slug will then move to a suitable environment, and it consists of pre-stalk and pre-spore cells, because it will eventually form a fruiting body consisting of a stalk and a spore at the end of it. So after this fruiting body is formed, as you can see on the diagram, the individual spores are released, restarting the life cycle of the amoeba. So some important factors for this model are chemotaxis of the amoeba towards higher areas of higher cyclic AMP concentration, ammonia inhibition, and thermotaxis and phototaxis, which exist biologically, but I haven't really implemented in this project. And why do we care about modeling D. discoideum? It's important because it's used as a model organism in biology. So in biology, we don't really experiment on humans very much. You'll use like C. elegans or a mouse or zebrafish or something else. And because because D. discoideum demonstrates very clearly concepts like chemotaxis, motility, cell signaling, and cell differentiation, we use it as a biological model. So hence it's useful for us to be able to have a computational model so we can kind of predict what's going to happen in this very important organism. The primary paper sources for this project are two by Murray et al., one from 2001 and one from 1999. The 2001 paper contains the qualitative results that I'm trying to replicate, along with the cell types, contact energies, target volumes, and other parameters. And the 1999 paper has the cyclic AMP and refractoriness ODE models, which are implemented. The project challenges. My big challenge this semester was a lot of vague explanations present in the papers that I decided to use. So my primary paper is the 2001 paper, in which the cyclic AMP model was not explained, so I had to go find that in the 1999 paper. Then there were other areas of non-clarity, such as whether to use the amount or concentration of compounds in the mathematical model, what the threshold for cell differentiation was, and how all of these different cells, these pre-stalk, pre-spore cells, should be initialized together. So now that we've established those challenges, let's dive into how I ended up conceptualizing this model. So the paper describes 5 different cell types: pre-spore cells, seen in green here on this diagram, 2 types of pre-stalk cells, a pre-stalk A cell, which stands for pre-stalk autocycling, and I'll explain what that means in a second, and a pre-stalk O cell. So the pre-stalk O cells are in red here, and the pre-stalk A cells are in dark blue at the top. There are also stalk cells, which will form the stalk of the fruiting body, and pathfinder cells. And it's kind of hard to see there in purple, the arrows pointing there. that help guide the downward growth of the stalk. As you can see here, the stalk kind of starts at the top of the model. There are also two extracellular components, a slime sheath, which will eventually surround the spore, and a stalk tube, which will surround the stalk. Additionally, there are two media. There is air and soil. In my implementation of the model, air is just the CompuCell 3D medium, and then we have a short little layer of soil at the bottom underneath our cells seen here. Now, how does this all come together? The pre-stalk O cells will differentiate into pre-stalk A cells. And the pre-stalk A cells will differentiate into stalk cells. And the rates of differentiation are dependent on how much contact area each cell has with the type it is supposed to differentiate into. The pathfinder cells, remember those purple cells from the diagram on the previous slide, they will guide the downward elongation of the stalk. The tube cells will eventually surround that stalk, and then the slime sheath will surround the fruiting body, the spore cells. And then the pre-spore cells just, they don't differentiate. They just eventually become the spore when it develops. Now getting into the mathematics, A cells, the autocycling pre-stalk cells, will produce an oscillating cyclic AMP signal, which is then relayed by the pre-stalk O and pre-spore cells. So the pre-stalk A cells, so the signal originates with the pre-stalk A cells, and the other cells, the pre-stalk O and the pre-spore cells, have to come into contact with that cyclic AMP in order for them to experience a spike in their cyclic AMP levels and then further pass on the signal. Now, the cyclic AMP signal is generated by the pre-stalk A cells, which are all the way at the top of the model. And the way we have chemotaxis implemented, cells will want to move towards areas of greater cyclic AMP concentration. So this would be the pre-stalk A, the pre-stalk O, and the pre-spore cells. So they will, as they move up towards the pre-stalk A cells, they all push up against each other as they move up. And this creates a pressure wave that will then drive the stalk growth downward. So between the cell differentiation of pre-stalk A cells into stalk cells and the pressure wave push, we end up with a net downward stalk growth. And then additionally, ammonia is supposed to inhibit cyclic AMP production. So here is the basic mathematical model for changes in cyclic AMP and refractoriness levels. So in these equations, C is the amount of cyclic AMP, R is refractoriness, and K is a constant. So the levels of cyclic AMP and refractoriness are dependent on these piecewise functions whose values change based on what the cyclic AMP concentration is at that moment in the model. So that is all implemented in antimony. So we can see that the way its model is currently written, the cyclic AMP, the dc/dt equation is always decreasing. Unless that value is negative, then it would be increasing. So now getting to a May 4th update and results. In order to help with the development of my model, the fruiting body model, Dr. Glazier had me attempt to replicate another model from an earlier paper that was just about the movement of the slug, the motile slug that I was discussing from earlier. So above you can see the figure from the paper of the motile slug as it moves across that x-axis. And at the bottom, you can see my simulation results. And initially, we had on the x-axis a block of blue pre-spore cells, a block of green pre-stalk O cells, and then a block of red pre-stalk A cells, all stacked on top of each other in rectangles. And these cells, they all have that cyclic AMP model that I described on the previous slide, or the previous two slides. And they are all supposed to chemotax towards areas of higher cyclic AMP. So eventually, this results in the slug crawling across the screen. So as you can see from this screenshot, the slug eventually moved upward towards the pre-stalk A cells, which were producing the cyclic AMP. However, the arrangement of the cells doesn't necessarily match what the model has. So we end up with an interesting case where we have some pre-stalk A cells on the side here, the red producing signals. And then we also have some extrusions of the pre-spore cells into the pre-stalk O cells, likely due to chemotaxis. So this model isn't perfectly tuned, but we have managed to show that proof of concept that we can get a slug to move. And on the right, you can see the heat map for the cyclic AMP levels. So the cyclic AMP, as it's relayed, will travel from the pre-stalk A cells up top down through the pre-spore cells. Now going back to the discoidium model, I have worked with Dr. Glazier to update the model such that now we have cyclic AMP waves visibly moving throughout the cells, which is the behavior we want. They're not necessarily the distance apart that they should be. Right now they're a bit close together, and the thought is that this impacts the pressure waves that we observe, so that if the cyclic AMP waves are too close together, then the pressure waves will cancel each other out, leading to no pressure to drive the stalk cells down. However, we do have a strong first pressure wave that does a good job of initially narrowing the stalk cells into a form that is more aligned with what we expect from the qualitative results seen earlier in the presentation. So that's promising. And then also cell differentiation is largely under control. Previously, I was having issues with cells differentiating too quickly, leading to this big mess of stalk cells that was uncontrolled. But now I've changed the model so that differentiation doesn't start until after the 80th time step, in order to give a chance for the stalk cells to narrow down a bit so they don't have too much contact area with the pre-stalk A cells that will eventually differentiate into stalk cells. And then from there, every 9th time step, there's a chance that the pre-stalk A cells will differentiate into stalk cells and the pre-stalk O cells will differentiate into pre-stalk A cells, with the additional caveat that the probabilities of differentiation are also decreased by a factor of 10. So in the cell differentiation mechanism, the random number generated to determine probability has to be lower than the contact energy, the percent contact area of the cell with the cell it's supposed to differentiate into times 0.1. And that was designed as a control mechanism so that cell differentiation wouldn't occur too quickly, though now I'm thinking maybe that I should let cell differentiation occur a little more to take more advantage of that strong first pressure wave that is coming down. So now I'm going to share my screen again so you can see some actual simulation results. We'll go desktop. All right, so ignore my PowerPoint in the background. So here we can see that this is mid-simulation around the 400th time step. The stalk cells are yellow, the pre-stalk A cells are red, the pre-stalk O cells are green, the pre-spore cells are blue. Outside is the slime sheet that's supposed to surround the spore. So now as you can see, the stalk cells have elongated a bit. The pathfinder cells are still surrounding them, still trying to find that path. And we can see a little bit of differentiation that's happening with some yellow spots among the pre-stalk A red. And what we would like to see is that as the pressure waves keep coming through due to the cyclic AMP, we should keep on seeing further elongation of the stalk cells. So here are the cyclic AMP waves. You can see they're pretty close together. and here's the pressure graph. So this dark blue curve right here, that's the first initial pressure wave that goes through with the first cyclic AMP wave. And now you can notice that the rest of these curves, though, you can kind of see them, they're significantly weaker. So the issue with this model then is that the first pressure wave is really strong, but after that, we don't have the strong pressure that will lead to that further elongation of the stalk cell. However, there are other promising aspects of this model. As the simulation runs for longer and longer, we do see the amount of cells grow taller and taller. Right now, we kind of start maybe closer to 100, but if you let this run for a long time, it will be higher. And yeah, so in conclusion, I think this project was a great exercise in trying to couple mathematical equations with qualitative results. I think that the vague descriptions in the papers were a contributing factor to kind of the slower motion of this project. But I think that there can be some fine tuning to get a model in which the stalk will grow completely down. Maybe that means I should be differentiating more from the get-go to take advantage of that pressure sweep. Maybe that does mean I need to play with the frequency, but playing with the frequency of the cyclic AMP waves to get them farther apart, that means that maybe I don't have the model that they're trying to replicate. If I try playing around with the ODEs or something, at that point, it's not replicating, it's changing the model to get some qualitative results. But overall, I thought I had a valuable experience with this. And I learned a lot about debugging and working with math and spatial models and considering how different factors contribute to different behaviors. I'd like to thank everyone for sticking along with me and having patience as I debugged my cyclic AMP model week after week and debugged my initialization week after week. And yeah, thank you. I didn't think this presentation was supposed to be too long. So that is where I'm at right now.
Well, I mean, one thing you might think about, again, I mean, we're done, so you don't have to, but one of the things the last time we talked, you plotted the region where the cells are activated and kivataxing and the value of the chemical field, the cyclic AMP, to check that the cells were seeing the gradient in the right direction.
Right.
That might be something you could do here to make sure that the cells are really being pulled inward in the way they should be. Another thing that occurred to me a little later, perhaps, was that the lambda volume that they use in the source paper is quite small. Now, if lambda volume is too small, the cells will be compressed and disappear completely.
Yeah, that's the issue I was having before.
But if it's too big, then they're going to have a harder time responding to the pressure. And so I wonder if part of what's happening isn't that the cells are a little stiff, that they're not, that if they were a little softer, the lambda volume were a little smaller, maybe they'd move a little faster. So those are two thoughts that I had. There is this issue that, and this is something it's not implemented in CompuCell, is that in the Pauline's
Am I frozen or is Dr. Glazier frozen?
I think it's him.
Okay.
Yeah, I think we may have lost Professor Glazier momentarily. Josh, I'm gonna need you to chime in because my dogs are about to go berserk. I won't be able to talk for a minute.
people still here?
Yes, I think everyone's still here. Professor Glazier should be back momentarily. While we're waiting for him, I want to say that you did an excellent job this semester. Oh, here's Professor Glazier. I'll just save my remarks and hand it back to him.
I guess my internet went out for a second. Seems that every time it rains, I lose my Wi-Fi. Go ahead, TJ.
So I think that Callie did an excellent job this semester. This was an unexpected exercise in reproducibility, or the lack thereof. And this is a very complicated model, and the target paper is brief to the extent of being very difficult to discern what all is going on. Fortunately, this work has spanned across a couple of papers. I think we tracked it all the way over the course of at least 8 years. This is definitely a reproducible model, but probably a lot more work than maybe we had initially expected. That being said, I think that we have a pretty strong grasp of why we're not getting the fruiting body. I think Callie can probably tell you more about that. But this is a cool model with a lot of details, and talk about persistence. She stuck with it, and yeah, hours and hours of debugging. So this was sort of a baptism by fire, and sort of, I would say probably something like the bowels of computational modeling. So excellent work this semester.
Thank you.
I mean, I think it would be fun. I mean, we should, whether you decide you want to do it or not, one of us should probably borrow that crawling slug simpler model and see if we can replicate that paper, which is got a lot of, as you say, you've got if you go back to your slide 11, it's doing the qualitatively right things, but the shape isn't quite right. And so there's issues with things like cell adhesion, and probably again with the stiffness of the cells that could be making a difference. And so there are also some what I'd call meta parameters, model meta parameters that we didn't really play with. We used the nearest neighbor spin update rule. We used, I think, fourth neighbor for contact adhesion. And I don't think that the papers of Murray told you what they used for those values. And changing those numbers can make a relatively big difference to the way the simulation behaves.
Yeah.
They shouldn't change the qualitative behavior, but they could change the quantitative behavior somewhat. And so I think maybe TJ and I should be, if you have time, we'd love to work with you at trying to get that, basically reproduce that picture at the top and see if we can make that work.
Yeah, we can discuss that. I mean,
Yeah.
This simulation, like it seems promising for sure. And I would have let it took a while to get to the point where it was crawling, not
Right.
crying. And I would have let it run for longer, but then I had to go back to this.
Right. But I know they built it. I mean,
Yeah.
it took Stan. Well, actually, that culmination paper was pretty amazing. The one you tried to simulate is pretty amazing. I think I mentioned this before. That paper was the reason CompuCell exists.
Yeah.
Because that I couldn't replicate that paper 20 years ago. And I said that if I couldn't replicate that paper, the likelihood that anybody else in the world could replicate it was essentially zero. Given that they're using a method I invented, if I can't do it, I don't think anybody else can. And so we wrote CompuCell because we wanted to make it easier to replicate models. But you've gotten really far in the sense that you've got the traveling waves there. They're very clear. You've got the movement of the tissue of those cells downward. And so I think structurally the model is there. The issues are going to be parameter tuning, and the problem is because the simulation takes so long to run, exploring the parameter space is really hard. This is the kind of case where probably you write a bunch of metrics to report on what's happening and to save movies. And then you throw it onto a cluster and run 400 copies at a time. They may each take 2 days to run, but at the end of a few days, you've got a few 800 parameter values. And then you can begin to explore the world that way. It's not an easy thing to do with these slow simulations. There was an earlier earlier paper, which was also very beautiful. It was a predecessor to this from somebody in the same group, which was by Nick Saville. Saville and Hogeveg. And
Oh, this one's Murray. Okay.
now this is Stan. So Saville was a student in that group before Stan. And he built a very nice slug model. His model was of the slug and its movement. And that would also be one that would be very interesting to replicate. Stan went on further after this paper and modeled the various kinds of mutants that could occur. There are different species of dictys, and they have slightly different shapes of their fruiting bodies. He showed how those worked as well. So I guess on your slide deck is great. The one thing I might do is actually show the curve of that piecewise linear function. So
Yeah.
on that slide. And then you mentioned the use of dictyostelium as a model for a model organism, which I thought was great. One thing to think about, one of the reasons dictyostelium was of such interest was because people who were interested in the origin of multicellularity found dictyostelium very interesting. So for humans, most of the time we're multicellular, except when we reproduce, when we're a single cell egg and a sperm.
Right.
Dicty have sort of the backwards life cycle, where most of their lives they live as single cells, and then they become multicellular just for the purpose of sporulation. And there is this apparent paradox, which is genetically all the cells in our body are very similar. They're not identical, but they're very similar to each other. And so you have what's called the disposable Soma hypothesis. As long as the germline reproduces, doesn't care what happens to the other cells. But in the case of dictyostelium, the cells that become stalk die in favor of the cells that become spores. And in principle, maybe if you're living in the ground, all of the cells near you are related to you, in which case that sacrifice can, through the price equations, be beneficial. But there's no guarantee that you are. And so this particular system apparently is very vulnerable to cheating. If you do a game theory analysis of it, nobody wants to become stalk because you die. Everyone wants to become spore. And so one of the things that was studied quite a bit, not in this paper, but later on, was what's the mechanism that regulates the choice of becoming pre-stalk or pre-spore.
That's interesting.
And they showed that if you took a slug which has a funny technical name of a grex. If you took the slug and you chopped off the pre-stalk region at the front, some of the pre-spore cells would redifferentiate to become stalk. And so there was some signaling that was going on to balance the number of stalk and spore cells. But essentially there was a mechanism where everybody's trying to become, they're all sending themselves a signal saying, I want to be spore. And they're all sending a signal to their neighbors saying, you become stalk. And so there was an interesting competition mechanism that regulates that, because there are species of dictee that don't make stalks where everybody becomes spores. But of course, then they can't disperse as well, because that tiny stalk may not seem very long, but it's long enough to get your spores away from the source. And so if you're in a bad local environment, it makes a big difference to the survival of the spores. And so this is a case where the sacrifice of the stalk cells clearly is beneficial to the ensemble of cells, but evolution never acts at the level of group selection. It has to be beneficial to the individual cells. And so there was a big mystery about how is it beneficial to the cells that die? And the answer has to be that it's beneficial to enough of your nearby relatives that genetically it's favored. And so there was a lot of very interesting work on the evolution of multicellularity, because in some ways, cancer can be thought of as a disease in which cells go from being phenotypically somatic, where they're going to die, to being phenotypically, at least some of them, phenotypically stem-like and immortal. And that's at some level a failure of some of the mechanisms that exist to enforce multicellularity. And so people like Loomis, who was a great dictyostelium biologist, spent many years thinking about how understanding the behavior of dictyostelium could help us understand the origin and maintenance of multicellularity. There also are species of bacteria which have somewhat similar life cycle. And it's quite interesting that there are eukaryotic organisms and also prokaryotes that have sort of similar life cycles. They also aggregate, they make slugs, they make stalks, fruiting bodies. And so the scale is smaller because the bacteria are smaller, but some of the same issues in terms of environmental variability, for example, show up in bacterial organization as well. So thank you very much. So let us know if you want to work more on this and how you'd like to, if so, how we can help you out. Okay.
All right, thank you.
Great. Thank you. So Drew, did you have an update you wanted to give us? You had a pretty complete talk the other day, but we're welcome.
Yeah, I could
Here you are.
I could show what I've done. However, I do have a problem. I need to see if you know what's going on. This just happened. I've not seen this error before.
Well, you got us on board.
So I don't know what happened, but if I grab a window, do you see how it's shooting off the screen, and I can't. If I try to drag this, it just flies away.
TJ?
Could you try switching to where you share your entire screen
Yeah, I can do that.
Yeah. Well,
I think it's a Zoom interaction.
yeah, Zoom is invasive in ways that I have not taken the time to understand.
Screen.
I know Zoom has a tendency to clobber Chrome. You would think that Chrome would be bolted down enough that that wouldn't happen. But I lose the edges of Windows when Chrome.
Yeah.
When I run Zoom.
I'm not really sure. I tried restarting the program, but it reloads the windows wherever they were when I last had it open.
And you're on a PC.
Yeah.
Yeah.
I'm usually on Linux, but I installed it for
Sorry.
Windows this time, and I don't know. Maybe I close out.
Yeah, if you want to reset the layout that's automatically loaded, you can go into the simulation directory and just delete sorry, I'm blanking. It's been a long day.
Josh, can you? I'm
Oh,
blocked.
okay. Yeah, I think I know what you mean.
It's the JSON file that specifies the layout. There really should be a one button. Do we have a one button clear of that in Player?
No.
There should be a one button reset.
Is it the underscore settings
Yes.
file? All right.
It seems that used to be even more of a problem because it used to be possible that you'd have that corrupted and then CompuCell would crash when you launched it. So it's
All right. Well, I still can't drag the windows though. I don't know what that is.
well, if you don't drag them and you hit tile, what happens? Under window, if you go to window and hit tile.
Oh, I
That
didn't know that was a
works.
button. All right, good.
That's probably the single most useful button in Player.
Yeah, I like that.
Although it doesn't exist in the Mac versions. It's a Windows only.
All right. Well, all right. So the things that I've added since last week, I said I was going to add a target concentration sort of slider so that whenever I've got it set to this right now, 0.001, which doesn't really mean anything, but for this simulation, I know that it's a good one to look at if I hit play. There we go. Is it going? There we go. So now you can specify target concentration and just see a different color represented on the histogram. It's really just a graphical thing. But I don't think I've added anything else. Oh, I did actually implement the delivery method thing. That was the other thing I did. So this is from, here, we'll look at the actual substrate there. So this is for IP delivery and it's coming from the outside. And if I stop the simulation and I switch it to like IV, oh wait, I forgot I gotta. There we go. Now it's coming from the inside. So I can actually change everything from the steering panel now. And I've also got an option for both. And I think one thing that's interesting is that I have yet to find any amount of settings that show IP delivery is better, and I'm pretty sure that the paper that I based this off of says IP is better. Although we did both agree that both delivery methods together works the best, but I found that interesting that I normally have IV is the better delivery system between the drugs.
I would have thought that would generally be the case. If the tumor is large and you give the drug from the surface, you wouldn't expect it would penetrate very deep.
Mm-hmm.
And so I would have thought IV would be better. I mean, that paper had some issues, right? We found some of the figures didn't make sense. And so it's possible there were some. Just one thing to always remember is that just because something is published in a journal doesn't mean it's right. And unfortunately, I can say that about my own papers. There are a couple of papers I published where I would have been much happier if the reviewers had caught my mistakes. People tend to be unhappy when the paper comes back from review and they want a lot of changes. But you'd sure as hell rather have the reviewer make the changes than have it published with the mistakes. So it's possible that there were some issues there, but this is very nice, and this could be a nice framework for going further for somebody, because you could add a model where you had cell death as a function of the drug accumulation. You could have recovery where the tumor cells regrow if the drug is not delivered anymore. You could have the evolution of drug resistance, where some of the cells are more susceptible to being killed by the drug than others. And so they're actually, once you have this basic structure where you have a vasculature that supplies potentially nutrients and drug to the tumor, you could do a lot. So I think this is actually a very nice it's a very nice model. I hope we can get it up on NanoHub, because I think this is a great demo.
Yeah, I just checked on my NanoHub tool. It doesn't seem that it's published yet. I'm not sure if there's something else I have to do, but yeah, I'm still
Josh,
waiting.
could you maybe meet with Drew and walk through that and make sure that you may have to issue a pull request asking them to make it public or something of that kind. Do you know, Josh? TJ, you must know how to get things up on that.
Oh, I usually pass that on to Giuliano.
Okay.
Now there is well, have you started the upload process at all?
I followed all the instructions from the one lecture we had, and I have it. If I look on NanoHub at the link that I have it just says I'm waiting for it to be published, I think, but I'm pretty sure I did that.
Oh, sure. Yeah, so you're so unless it's requesting feedback from you, then you're waiting on NanoHub to push some buttons. Now with that same interface, you can use that to publish revisions, and it just walks you back through the same sort of process. So all of the testing and uploading and so on and so forth. So I'm not sure if you can post a revision before publishing. That may be a case of deciding whether or not you want to cancel the current process and just start over with your revision, or if you just want to wait until after it's published and then start the process again with a revision. But that site, that interface, that's your go-to place for being able to manage each of your tools.
one thing is to make sure that they're not waiting for you to ask them to publish. How long has it been sitting in this waiting to publish mode?
I'd say it's been at least a week.
No, then it's probably waiting on you. I don't think they usually take that long, do they? DJ?
It's been known to happen, but
Or maybe send a reminder to them about it.
yeah, I mean, you can tell from the interface. It'll tell you if it's waiting for feedback from you. It should say in pretty bold font on the right-hand side of the screen.
If you screen share, we could look at it now, see if there's
Yeah.
anything. And if nothing else, we could always generate a message to them saying, please publish.
All right. Yeah, this is what I see, at least on the right.
Yeah, so this is the landing page. Yeah, there you go. Now in the top right, see how it says tool status? Let's see what that says.
Hmm. How do I get to the publish?
So
area?
it looks like there may have been some steps in the process that might have been missed. Yeah, because this, at least the tool that we're looking at here, this is pretty far from being published. Was there a different, you know, were you seeing something different as far as like the status of the tool you've been working on?
What do you mean?
Like, did it look like it was approved and nearly published, and now it says that it's registered, and that's...
Well, I think one thing that was different is that whenever I was first like uploading this to NanoHub and stuff, I stopped like halfway through the process, and when I came back to it, it was different, and I don't know, I can't really figure out how to get back to whatever flow it was on to publish it.
Because this says you don't have any source code.
Yep.
This says it's not uploaded.
That is weird, because I think it did before, now that I think about it.
But somehow it's lost your code. So you have to re-upload the...
Yeah, we may have to work on this maybe offline a little bit. This might take some debugging.
Yeah. Is this what the GitHub is at least supposed to look like?
Let's see. Yeah, that looks like the correct overall structure. Yeah, so this might take a little work offline, but yeah, it looks like there's an issue with getting your code. Yeah, actually getting your code uploaded.
Yeah, I don't think that changed anything.
Josh, are you there? Have you walked through this process?
Yeah, I'm
No.
here. I'm just having a little bit of troubles with the internet.
No, that's fine. Just it seems to not have. You have to do a finalize or something like that.
Right. You're just waiting for them to release it. So
No,
once
but
you
it's-
submit
it-
it, you have to
it's-
wait. Yeah, it takes like 1 or 2 days for them to release it.
No,
you submit.
but it seems to have, it doesn't have his source code. It's lost his source code.
Oh, right. So yeah, Giuliano is the one that knows a bit better how to deal with these problems. But once you have selected the Git URL, they're supposed to be able to put it from there. But I guess they are just having, yeah. Did you get an error message?
I don't think so.
Because usually what happens is that you get an error message telling you that something was wrong with your code. But yeah, Giuliano is the one who knows much better how to deal with these problems.
Can you please send an email to Drew and Giuliano to make sure they meet tomorrow to get this taken care of so Drew can finish?
Yes, of course.
Since his project is all done.
Right.
And works very nicely, it's not fair to him that we can't get it up on NanoHub.
I agree. Well, I'll send Giuliano.
Because I think it's a very nice simulation. It's perfect, and you've used the sliders interface box very nicely. I think it's a great job. So I'm sorry you're having some frustration about getting it uploaded because it seems to be a great project.
It's fine.
Okay, well, I guess Jeffrey, you're the last one for today. You have an update for us?
Yeah, I can show something. So I have this plot still, and I'm getting this timeout error for the message rate, and I'm not sure how to change it on NanoHub because this looks like a terminal command that you would use for running Jupyter offline.
I looked into that. It turns out that there is a bug with an older version of Jupyter Notebook Server, and that basically in the command line that launches Jupyter Notebook Server, we need to add that argument, or they could upgrade to a newer Jupyter server.
Okay.
I looked into it. Basically, it's a common problem with a particular version.
Okay. So yeah, I have then this Puisé flow simulation that seems to work pretty well once it reaches the steady state, but it's not here. And then you'll be able to toggle on and off potential boundary conditions, which we've talked about earlier. And so then this will be one notebook, and I also have this one, which is the same thing, except it has this little chemical ball in the middle. I think I'm having internet connections too because it's not connecting to the kernel right now. But yeah, so then this is going to show there's a plot for the chemical species as it, you can see the plot, the concentration. Except right now it's not working in Jupyter. It works when I run it from the terminal, but the plots are again not showing up. So I'm still trying to figure that out in Jupyter. And it's not the same issue as before because this is the plot here. So I'm still I guess, trying to look at that. But I think Andy and I talked about this, focusing on these two for the project, and I think we're planning to keep talking and maybe work on another paper later on, which will focus on the fluids and include some of the other simulations, because I think I'm running out of time to do notebooks for all of the things I've made right now.
Right. You've done a lot. Everybody's done a lot. I certainly don't want to punish people for technical issues. People have worked hard. Okay. Is there anything else anybody would like to go over this evening? Okay, well, thank you, everybody. I appreciate you showing up in the middle of exam week. And I guess technically you have to tell me if you, I mean, people have done a lot of work. If you want to be graded on the work you've done so far, that's fine. If you want an incomplete and you want to have a little more time to work on things, let me know either way. Just send me an email and I can set it up either way for you. I want to make sure that everybody has the opportunity to get the things that they care about the way they want them. And I guess there may be some homeworks, if there were homeworks people wanted to do that they were planning on submitting, please get them in, because we need to get those graded by the end of the week to be counted. If you're not doing, if there are homeworks you don't want to do, that's fine. I mean, it's not, the world is not going to end for one or two unfinished homeworks. But if there are homeworks you had planned on finishing, please let us know. Okay, well, thank you everybody for showing up tonight. Appreciate it. And I'm glad that people have been sticking with it. And I'll be available to talk to people about projects, and if they'd like to at any point. Okay. Josh, is there anything else you wanted to say before we break? No. Okay, well then, thank you very much. Everybody get some rest, and good luck with your exams and other courses. Okay. Good night.
Thank you.
Thank you all for sticking it out. Okay, bye. Sorry, Andy, did you have something you wanted to say?
Oh yeah, I got all.
Hold on.